So far, we have looked at how we can predict properties of proteins and of small molecules. However, we didn't consider yet how we can make predictions for properties that involve both a protein and a small molecule. In this video, we are looking at how we can predict different types of interactions between proteins and small molecules. As I said, we are now interested in predicting different kinds of interactions between proteins and small molecules. This means we want to use information about the protein and the small molecule, and we want to be able to predict in which way those two molecules interact. But why is it even relevant that we can predict such interactions for proteins? On the one hand, proteins, in particular enzymes, play a crucial role in cellular metabolism, and hence knowing the function of enzymes is important in medical and pharmaceutical research for the study of metabolic diseases and drug development, and this requires knowledge about the interaction of proteins and small molecules. On the other hand, enzymes play also an important role in biotechnology and bioengineering, for example, for the synthesis of drugs, chemicals, and biofuels in food processing or in the degradation of waste materials. For these tasks, it is important to find proteins with specific properties that are suitable for processing or transporting specific substrates. However, for the vast majority of proteins, their functions as well as their kinetic parameters is unknown. To overcome these limitations, it is desirable to develop prediction models for predicting protein-small molecule interactions, for example, for predicting potential substrates of enzymes or transport proteins, for predicting the Michaelis constants Km of enzyme-substrate pairs, which describe the binding affinity between an enzyme and the substrate, the turnover number kcat of an enzyme catalyzed reaction, describing the maximum catalytic speed of an enzyme, or for the inhibition of a small molecule drug for a target protein. The reason why this is interesting is that when developing drugs, a common scenario is searching for a small molecule drug that inhibits the function of a target protein. On this slide, I'm showing you a typical prediction pipeline for those prediction tasks that involve protein-small molecule interactions. If we want to predict an interaction between a protein and a small molecule, we need a numerical representation of the protein and a numerical representation of the small molecule. We have seen previously how we can achieve this. For representing the protein, we can use pre-trained protein transformer networks with the protein amino acid sequences as the model input. And to generate representations of small molecules, we have seen that we can use small strings to calculate numerical representations using either traditional fingerprints, like the ECFPs, or graph neural networks, or we can use transformer networks. Here I listed graph neural networks as an example. Since we want to have a single numerical vector containing both protein and small molecule information that we can use as the input of a machine learning model, one way of achieving this is by simply concatenating both vectors that were generated by using two separate deep learning models. This vector can then be used as the input for a machine learning model that is trained to predict the protein-small molecule interaction of interest. We've already discussed that gradient boosting decision tree models are typically well suited for such prediction tasks. This protein-small molecule interaction task can be either a regression task, such as predicting how much a small molecule drug inhibits the function of a protein, or it could be a classification task, such as predicting whether or not a small molecule is a substrate for a given protein. If we look at this process again, we see that we first separately generate numerical representations for the protein and the small molecule and then combine both information in a concatenated vector. We see that this means that we have no information exchange between protein and small molecule until after the numerical representations are generated. However, this might be a limitation, and we might be able to generate more meaningful representations if we allow the model to look at the other molecule for which we want to make a prediction during the whole process of generating a numerical representation. The reason why this is difficult, or at least not obvious, to achieve is that the protein and the small molecule are typically represented on different scales. We have seen that proteins are typically represented through their amino acid sequences when generating protein representations, and small molecules are represented on a much more detailed level by representing each atom and bond in the molecule. To achieve a model that can represent both modalities, the protein amino acid sequence and the small molecule SMILE string in the same input sequence, it would be desirable to use a transformer network that can process the amino acid sequence of the protein and the SMILE string of the small molecule in the same input sequence. To achieve this, we can look at transformer model architectures that have been developed in the last couple of years for processing different types of model input. For example, such models are capable of processing both images and text in the same input. For example, we could have a transformer network that can take both an image and a text as the input, and we could predict whether those two input modalities fit together. This could be, for example, interesting when we want to search an image database for an image that fits best to a specific image caption. There exist many different options to achieve such a model, and we're briefly going to look at the broad ideas for 3 of these here. In the following figures, the tokens of the 2 different modalities are represented by purple and green. As we have seen on the previous slide, those 2 modalities or input types could be, for example, image and text, or protein and small molecule. One option of doing this is simply concatenating both input sequences and using the resulting sequence as the input for a single transformer network. This requires that we tokenize both input types using the same vocabulary. This means that we have separate tokens for each input type, but they are stored in the same vocabulary and used as the input for the same embedding functions. This allows the transformer network to look at both input modalities simultaneously. A related option is to first pass each input type through a separate set of transformer network layers before using the embeddings of both sequences as the input of shared transformer network encoders. The advantage here is that we allow the model first to generate good representations of the tokens of both sequences before we allow the model to look at the other input type. It might be easier to extract useful information from the other input type once we already have meaningful representations. And the last option we are shortly discussing is using cross-attention between both input sequences. This means we have 2 separate sets of transformer layers, but for calculating the attention scores, we are using the embeddings from the other modality. We know that we have 3 types of inputs to our attention function: queries, keys, and values, in the image represented by Q, K, and V. And typically in our encoder, these are the same and are equal to the embeddings of the input sequence. But in cross-attention, we are also using the embeddings from the other input sequence to calculate the attention scores. This means we use the embeddings from the other input sequence to update the token embeddings of a sequence. This has, however, the disadvantage that we are not using any self-attention anymore. And we can, of course, also have completely shared layers on top of these cross-attention layers, or we can combine these different types of multimodal transformer blocks in many different ways. We are now looking at the Prothmes transformer network encoder that allows to process both a protein amino acid sequence and a small molecule SMILE string. And the architecture of this model resembles most closely the second architecture type that we've seen on the previous slide. We first tokenize both sequences separately and pass them through the ESM1B transformer network and the Chamberter2 small molecule transformer respectively. This gives us meaningful embeddings for all tokens from both input types. These tokens can be mapped to the same embedding space with the same dimension using linear layers. Since we then have embeddings of same dimension, we can use the amino acid tokens and the small molecule tokens as the input of a transformer network encoder. The attention mechanism of this transformer network now allows to look at the small molecule while extracting protein information, and vice versa to look at the protein while extracting small molecule information. This transformer network can be trained for protein small molecule interaction tasks of interest, for example, for predicting whether a small molecule is a substrate for an enzyme, or for predicting how much a small molecule drug inhibits the function of a protein. For these prediction tasks, we have a classification token that you can see on the very left that we can use as the input for a fully connected neural network in the same way as we have seen for how we fine-tune pre-trained transformers. After we trained this model for predicting a protein small molecule interaction, we can use this as our final prediction model since we trained it already for the task of interest. Alternatively, we can also extract after training for each molecule the updated classification token embeddings. This vector is now a task-specific representation of the protein small molecule pair because we trained the network to store all information that is relevant for the prediction task in this vector. We can use this vector as the input for an alternative prediction model, such as a gradient boosting model, which we train for the prediction task of interest. This typically leads to slightly improved model performance. One challenge that might arise is that we have no negative data to train a binary classification model. Imagine that we want to build a model for predicting substrates for enzymes. We remember that substrates are the small molecules that are converted by enzymes into different small molecules, the products. And each enzyme has only a very limited set of substrates. We could achieve a model that predicts substrates for enzymes by building a model that takes an enzyme and a small molecule as its input and predicts whether the small molecule is a substrate for the enzyme or not. So we have a binary classification task. To train such a model, we would go to protein databases, search for experimentally validated enzyme substrate pairs, and we would be able to find many thousands of such pairs. However, since we want to train a binary classification model that either predicts the positive or the negative class, we also need negative data points. That means enzyme small molecule pairs, where it is known that the small molecule is not the substrate for the given enzyme. Unfortunately, such data is in general not stored in large protein databases, and thus sometimes we have to find a different way to get negative data points. On the next slide, I will talk about how this might be achieved. Let us further consider the example of our enzyme substrate prediction model. As I said, in the literature and in protein databases, we can mostly only find positive enzyme substrate pairs. So how can we get the negative training data? If we compile such a dataset with positive data points, we have a few hundred up to a few thousand different small molecule substrates in our dataset. And let's imagine all of these substrates are displayed on this slide, where more similar molecules are closer to each other. For every given enzyme in our dataset, we have one or multiple true positive substrates, here displayed in green. And although many enzymes can be promiscuous, which means they can have secondary substrates for which they did not evolve, but to which they can still bind and catalyze, among this limited set of small molecules in our dataset, enzymes are quite specific towards their substrates. This means if we randomly pick small molecules from this set, there is a very high likelihood that these molecules are not substrates for a given enzyme. So what we can do is we can, for example, randomly select a few, for example, 3 negative small molecules for every positive enzyme substrate pair. The number of sampled negative data points for each enzyme can be treated as a hyperparameter. Generating more negative data than we have positive data not only provides us with more training data, but it also more closely reflects the true distribution of positive and negative data points. However, when doing such a completely random sampling, we might find that the trained model is not very good at distinguishing between the true substrates and small molecules that are structurally similar to the true substrates. To overcome this, we can only consider those small molecules that are structurally similar to the true substrate. This results in a more difficult learning task that forces the machine learning model to learn the important features for distinguishing substrates from structurally similar non-substrate molecules. To summarize this approach, when we are missing negative data for prediction tasks of interest, we can check if we can easily infer negative data points without the requirement of experiments. For many biological prediction tasks, this might be the case. It might be that if we make any random input, for example a random enzyme small molecule pair, that the likelihood that this is a negative data point is super high. In these cases, we might simply generate negative data points. This data generation might lead to some false negative data points that we use for training. However, if the chance is low that we create many of these, this is probably still fine and will not affect model performance too much. If we decide to randomly sample negative data points, we have different options for doing this. We can do this completely random, but this might not result in a prediction task that is difficult enough to learn the important aspects for classifying data points as positive and negative. So we also have the option to sample negative data points that are similar compared to the positive data points, such that the prediction model really has to learn what the important aspects are for distinguishing positives from negatives. How similar the negatives are compared to the positives can be also treated as a hyperparameter. And finally, we can decide how many negative data points we want to generate. We could, for example, treat the positive to negative ratio as a hyperparameter if we have a validation data set with experimentally validated positive and negative data points, where we can evaluate the effect of this ratio on real-world experimentally validated data.